Now we can start. I want to welcome everybody who's joining us this morning or afternoon. My name is Sofia Rodriguez. I'm the Director of Academics for Tropical OTS, Organization for Tropical Studies. I want to welcome everybody to this session. It's session number 5, Emerging Frontiers in Tropical Ecology, Voices from the Next Generation. With this symposium, we want to highlight the research of the newest generations of tropical researchers that have recently obtained awards from ATBC or the Organization for Tropical Studies, OTS. Both OTS and ATBC have provided great research opportunities for many generations of researchers. And so we want to emphasize the importance of these new generations in building new knowledge in different areas within tropical ecology and conservation. ATBC, together with Biotropica, has a very well-known awards program for recognizing outstanding papers published in Biotropica by young investigators. Also in 2020, ATBC started a new grants program, ATBC Seed Research Grants, to promote research for the earliest generation of researchers. OTS is also renowned for its graduate and undergraduate courses, for providing research fellowships for a new generation of scientists as well, to conduct their projects at our research stations in Costa Rica and South Africa. And we also have an awards program for recognizing young investigators with outstanding publications. We really hope that in the long term, we'll be able to continue doing this symposium annually. So we're going to start. I want to remind you to submit your questions for the speakers at any time during the presentation, but do so at the Zoom Q&A button. Don't do it in the HOVA function. And also remember to say to which speaker you are addressing your question. So today with us, we have some investigators who have won the ATBC Seed Research Grant in 2020. These are, and I apologize if I'm mispronouncing anyone's name, Hayden Davis, Jacob Motowama, George Hernandez Corrales. We also have Scheherazade, who won the Peter Ashton Prize 2020 from ATBC, and Benton Taylor, who won the OTS Outstanding Student Paper Award in 2017. And now we will begin our presentations.
Hello all, thank you so much for coming to this talk. My name is Hayden Davis and I'm a graduate student at the University of Washington. Before getting started, I just want to thank my co-authors and my funding sources that have helped this project come to fruition. So, and in particular, I'd like to thank the ATBC for one, hosting this symposium and inviting me to give a talk, as well as for providing funding that has allowed a lot of this work to move forward. So my work focuses on establishing species boundaries in Southeast Asian geckos. And in particular, I'm focusing on this genus Cerdidactylus, which is the third most diverse vertebrate genus in the world. But if you look at the species trajectory, so this is the number of species described through time, you'll see this recent explosion of species where we're going from 77 in about 2000 to over 310 today. And what's a little bit concerning about this is that a lot of these species are being described with really limited evidence. So for example, a lot of them will have a lot of taxonomists will use these arbitrary cutoffs of say 3 to 5% in a mitochondrial marker, and then use a posteriori methods to look for morphologically diagnostic features. And also what's interesting or potentially concerning is that with over 310 species described, there's only been 8 that have done so using any genetic data other than mitochondrial. And so this brings me to my two questions of this research, which is how much, if any, gene flow is ongoing between putative species. So between these recently described or species that maybe have that 5% mitochondrial cutoff, how much gene flow is there that's currently going on? And so I put this in a framework of the speciation continuum. And this is the continuum wherein you're going from one species and then eventually having a diversification event into 2 species. But in the midst of this, you have this gray zone of speciation. And this is where it's unclear where population boundaries end and species boundaries begin. So it's possible that with these low mitochondrial divergences, we're looking at something that's in this very early stage of this diversification process. And secondly, is and really relatedly, is whether or not single locus data is sufficient to delimit species, and in whether or not those patterns that we see for delimitation are matched by the genomic data. So to get at these questions, I focused on the Southeast Asian island of Borneo, shown here in the middle of Southeast Asia. And this is a great study system because it has the Cerdidactyls on the island have comparatively low recognized diversity, but they have a high amount of genetic diversity. And this recent paper really exemplified this. So this blue, yellow, and green group, these were all three considered to be conspecific until this paper came out, despite upwards of 22% divergence in the mitochondrial gene ND2. But further, if we look at these colorful bars on the side, these gaps in the middle, or sorry, these are species delimitation models, and these gaps in the middle indicate potential species that warrant recognition. And so current methods for many taxonomists involve getting single locus data sets from mitochondria and then using these species delimitation models to look for these splits and then going retroactively back to look for morphological characters to confirm that. And so this really sets this ideal framework for testing the validity of this method. So to do so, let me move this a little bit. To do so, I gathered morphology data using both meristic and morphometric data for all species in Borneo, and then have recently gathered genomic data using ddRADseq and shotgun sequence genomes. However, I've not been able to complete this data set because that's going to require that I get back to Borneo, which due to COVID has not been able to happen. But using the data that I do have, I've conducted population and species delimitation analyses to look for gene flow patterns, for population structure, as well as just looking at the multi-species coalescent processes. And this is all to really try to figure out where in this continuum these lineages occur. And what we're seeing is that there's certainly high diversity, and this has actually led to the description of 4 species, which are either really morphologically distinct or have been really genetically distinct. But what we see when we look at the RADseq data, so this is the admixture plot and the PCA for 2 different species complexes, species complex 2 and 1. And what we see is that it's not so clear cut what these boundaries are that are being depicted by the mitochondrial data. And instead, you kind of get these gradients of diversification. But what we're also seeing that's a little concerning is that there's conflicting signals between the genomic and mitochondrial data. And there's 2 scenarios I want to point out. So the first being all these dots here are from one locality that's isolated on a mountaintop. But when you look at this with the mitochondrial data, then you start getting these other individuals from a geographically disjunct population that are appearing with this group here. But when we look at this with genomic data, it's very clear cut that this is the only group. Secondly, we see the same pattern here where the species delimitation models for the single locus data of mitochondrial data sets show that there's 2 distinct species. But when we look at this with the genomic data, we again see that that's not the case. So are the current practices over splitting gecko lineages? We don't really have enough evidence to confidently say this, confidently say this, but what we do see is that the mitochondrial data would have, in these 3 species, would have led to the addition of 2 additional species in comparison to genomic data. But what's more concerning is that it seems that a lot of this diversification that's being reported is really the product of isolation by distance. And we see this again with Borneo Highlands. This is the isolated mountaintop lineage that I was mentioning. And then you have this whole broad range for this orangish red group here. And when you look at the mitochondrial data, you start to see these delimitation models kind of splitting off by geographic locality, but that just is not supported by the genomic data. So even with all this data, it still comes to the question of how do you decide how a lineage should be elevated. And one thing which is a very foundational and a preliminary thing that could be done is looking for a reference taxonomy. So this, for example, Muluensis and Hantu, these are two species that are ecologically and morphologically extinct, and by all means would be considered a species under even the most conservative species concept. And so you can use this cutoff for this pairwise distance between these two as a reference for a bare minimum of how much is necessary. Second, you can take into account effective population size, especially when dealing with genomic or multi-locus data sets. This can be very foundational. Assessing whether there's ongoing gene flow, and if there is there any other feature that could be used to diagnose these species? It's a confident metric that's reducing their gene flow patterns. And lastly, do different data sources support the same conclusion? So do you have morphology and mitochondrial data saying the same story? Does that genomic data support that same story? Looking at these from multiple components is really helpful. And so why does this all matter? So species are really the foundation of any conservation process, because if we don't understand the diversity that's present in a given area, arguing for that protection can be much more cumbersome. But hastily describing species based on these mitochondrial or arbitrary delimitation cutoffs can also really both undermine the value of what a species is, as well as undermine the validity of taxonomy, which has already been under pressure from funding sources and other bigger institutions. And this isn't to say that all species descriptions, or even most, should require genomic data. But what this study aims to do is really assess whether the methods that we're currently using are accurately documenting species diversity, or if we're looking more closely at population or genetically anomalous situations. And so what I hope through this is that it can help to establish a standard for preliminarily geckos, but also on the broader scale, just more rigorous methods for documenting Earth's biodiversity. And with that, I'm more than happy to take any questions that you all have, and I look forward to hearing from you.
Hi everyone, I'm Sarah from Indonesia. I'm representing my organization called Progress. This is a local-based NGO in Luwuk, Central Sulawesi. In this opportunity, I would like to present about our works on studying and trying to save flying foxes, which are the largest fruit-eating bats in the world, and they also distribute in Indonesia, including in Sulawesi. We started our journey with studying the threats that are faced by the flying foxes. So they are the most commonly hunted species and sold for bushmeat markets in the northern part of the province. And to satiate the demand coming from this province, hunting actually occurred throughout Sulawesi, where the southern part is the major supplier. And we estimated about 500 tons of bats are annually transported and sold for bushmeats. So it highlights the urgency of saving the flying foxes given the high level of threat that they face. But we also figure out that there's a challenge in terms of conserving them, because first, there's a lack of protection status for many of the bat species. And second, they usually distribute outside protected areas. And the third one is basically there's a lack of scientific research on bats. So we decided to start with a research on ecosystem service provided by flying foxes, with special attention to durian pollination service, because durian is most loved in Indonesia and particularly in Sulawesi. It is also the very important fruit community. And the second, we also look at the seed dispersal and pollination service that are provided by the bats for other Sulawesi native trees. And this is actually the paper and the reason why I'm at this symposium in the first place. This is the one that I published in Biotropica and won the Peter Aston Prize for the student outstanding paper. And on this research, we conducted pollination exclusion experiments, which some of the durian flowers we cover, and some we are allowed visitors vertebrate, including bats, to visit. But to know which species, we set up camera traps. And along for over 2 months, we counted the durian fruit stats for each of the durian inflorescences that are under the experiments. And from these entire experiments and also the camera trapping setup, we found out that bat visitations, particularly the flying foxes, resulted in a higher durian fruit stats. And we counted the total productions of the durian in the village and also in the district, estimated the dependency ratio of the durian flowers on bats using the data from the experiments, and multiply them with the local price. We found out that the bat pollination service to durian are worth $117 per hectare. And it shows that this bat pollination service is important for this economically valuable fruit for the local community, but also culturally valuable, because eating durians is something that people looking forward to every single year. And you can look at and find out more about our research on this paper. And after the durian, we continue our journey to explore the ecosystem service provided by bats at different areas in Sulawesi. We collected their fur and identify which plant species that get the benefit. And we found that the bats provide seed dispersal service for many of the hardwood trees and also pollination service for mangrove. So these research are actually become the foundations and our motivation to establish an NGO that I mentioned before called Progress. We picked one of our research locations in Central Sulawesi in Bualemo to start working together with the community to protect the flying fox roost with that has 3 different species that you can see in here. Our organization visions is actually to initiate and empower locally led conservations, particularly the youth. We expect them to later lead and run the protections for threatened yet overlooked species in Sulawesi. And bats are actually the start. And we did a lot of activities. In the prior conditions, we saw that the local community actually allowed the hunters to take the flying foxes. So we work with them to develop a sense of ownership to these flying foxes and how they can realize and notice that from the water and the air that they actually enjoy from the forest also get benefit from the flying fox service. And also the durian open the people's heart to more likely getting interested in bats. But over time, since 2018, from our activities from capacity building, population monitoring, awareness campaign, and community engagement, we successfully stopped the hunting of the flying foxes and saw 76% increase in the maximum number of bats roosting on the island. We realized that the fluctuations of the bat populations are actually natural. And the community sites, there are children and youth, hundreds of them also join our awareness campaign and capacity building activities. We are very excited because from the bats, we learn a lot how to address the challenges to work on these not so charismatic species. So we are expanding to other species, threatened yet still overlooked. There's the Forsten tortoise, and then we're also going to expand for the Talaut cuscus in the next few months or even few years. So wish us luck. This is part of our other works, and I hope you can enjoy our journey to learn about the bats and try to save them and expand our work. Thank you so much, especially for our support and also from the government of Central Sulawesi. And please follow us and check out more of our works. Thank you.
Hi, everyone. My name is Jacob Mutuama. I'm a PhD candidate in the Department of Ecology and Evolutionary Biology at the University of Tennessee, Knoxville. I study population dynamics, ecological interactions, and ethnobiology. Today, I want to talk about how nitrogen availability in soil mediates the effect of distance from climatic center on herbivory rate for a tropical plant. The center-periphery hypothesis predicts that species are more abundant at the center of the distribution and decline toward the periphery. It is therefore expected that herbivory rates will be lower at such center than at periphery, explaining expansion limitation via negative influence of biotic drivers. However, if the center of the geographic range coincides with the preferendum of the species, one will predict high herbivory rates at the center than at periphery, consistent with the resource availability hypothesis. Previous studies have treated these two hypotheses in parallel, limiting our understanding about how herbivory could drive range limitation. We conducted our study in Benin, the country right here in West Africa, and we sampled 12 populations of Tomeja atacoensis across the entire range of the species. And the species is eaten by two types of herbivore. The first one is Phyllodes costivillatrix, which is this caterpillar, and the second one is Phyllopoda abriensis, which is this beetle. In every single population, we install 5 plots, each plot measuring 24 meters square. In each plot, we collected soil data and we did nutrient analysis. We also collected light availability in every plot, species diversity, like phylogenetic diversity, interspecific competition, and every single individual records. Finally, we use leaf bites to measure herbivore rate for every single individuals. We also use the occurrence record of each individuals to combine those occurrence record with bioclimatic data, and we build this minimum volume ellipsoid, which represents the niche of the species. For each population, we calculate the distance from each population to a hypothetical center of the minimum volume ellipsoid, and we call that distance the climatic distance. We combine all these variables to build this meta model, where we assume that abiotics factors here, such as soil phosphorus, soil moisture, soil nitrogen, soil pH, soil potassium, and light, will decrease from center to periphery, consistent with the center-periphery hypothesis. We also assume that herbivory rate will increase with soil phosphorus, soil moisture, soil nitrogen, soil pH, soil potassium, and light, consistent with the resource availability hypothesis. So let's assume that beta 1 here represent the slope of the relationship between distance from climatic center and soil nitrogen, and beta 2 is the slope of the relationship between soil nitrogen and herbivory rates, and beta 3 represents the slope of the relationship between distance from climatic center and herbivory rates. If the product of beta 1 and beta 2 is less than beta 3, it means that the direct effect here is stronger than the mediation effect. And this was our goal here by doing this meta model to understand how descent from climatic center could affect herbivore rate via mediation of abiotic or biotic factors. And we use structural equation model to estimate this coefficient in R. We found that soil nitrogen decreased from center to periphery, consistent with the center periphery hypothesis. And we also find that herbivore rate increased only with soil nitrogen, consistent with the resource availability hypothesis. And we conclude that soil nitrogen mediates the effect of descent from climatic center on herbivore rate. Our study shows that the low level of nitrogen at the periphery does not allow the species to compensate for the lost tissue, and this may explain why the species cannot go beyond a certain limit. I would like to finish my presentation by thanking the Association for Tropical Biology and Conservation, ATBC, for providing the seed grant to conduct this study. I would like to thank the National Institute for Mathematical and Biological Synthesis for providing a fellowship for my graduate study, the University of Tennessee, Knoxville, also for providing funding to do this research, and finally the British Ecological Society. I would like also to thank my two guides and field assistants. Thank you.
Hello, I'm Georgia Hernandez. I am a PhD student at the University of Connecticut. Today, I will talk about the temperature limits of tropical plants and how it varies along an elevation gradient. The projected increase in temperatures in the next century is between 1.5 and 6 Celsius, and it's expected that tropical plants are going to be particularly vulnerable. Part of this reasoning lays in the hypothesis that tropical organisms have narrow thermal tolerance and have a constrained adaptability to high temperatures. This hypothesis was displayed by Daniel Jensen in his famous paper of 1967. The logic behind this classical hypothesis is set on an OTS course. This ecology course comprises students from all around the world that come to Costa Rica. And here is going to be represented by a bear. When Jensen was given this course back in the 60s, he noticed a pattern among the students from the US and the TA that was from Costa Rica. Here represented by Slav, a new national symbol in Costa Rica, by the way. And Jensen noticed that when the course went to a field site in the lowlands of Costa Rica, a very humid and hot place, the US student was wearing the same kind of outfit, just a shorts and a normal shirt. But the TA was wearing shorts and was very sweaty and hot. When they went to another station in the mountains, again, the US student was wearing the same kind of outfit because he was just fine with the weather. However, the Costa Rican TA was so cold, he was wearing a thick sweater and a beanie and closed shoes. So Jensen was curious why the TA that was from Costa Rica was having a lot of trouble getting to his optimal temperature, while the US student was very comfortable in this range of temperatures. So this is where the famous paper of Why Mountain Passes Are High in the Tropics was born. The US student was used to endure all 4 seasons back in the US. That included a large temperature range compared to the Costa Rican temperatures. Meanwhile, the TA was from a specific region of Costa Rica, and he was used to a very steady temperature range all year long. Well, a shorter version of this story is that narrow temperature ranges, as we experience in the tropics, will select for organisms to have narrow temperature tolerances in local populations. This applies to ranges in a latitudinal scale, or along an elevation gradient. This is reinforced in the tropics because of the narrow temperature range compared to the temperate zones, where by every 100 meters you go up in a mountain, temperature decreases by 0.6 Celsius, which is very dramatic decrease in temperature for such small variation in elevation. Based on Janssen's hypothesis, I wanted to know if there is local adaptation in heat tolerance along an elevation gradient, and use the Symbiotic as my study model. Then we can also ask if plants are adapted to future temperature conditions under a global warming context. We can measure how stressed a plant is by using a fluorescence parameter. Here I used FB over FM, and thanks to this measure, we can then determine heat tolerance in a very specific way with how the photosystem 2 is affected. In a previous study, it was demonstrated that there is a phylogenetic signal for heat tolerance in a species of Symbiotic from the lowlands of Costa Rica, where closely related species tend to have similar heat tolerance than in species that are far apart in the tree. Based on this, I wanted to answer 3 questions. First, if heat tolerance here at T50 and thermal safety margins are locally adapted to life zones along an elevational gradient. I'm using thermal safety margins because it is a good tool to understand plants' vulnerability to climate change. For the second question, I asked if heat tolerance is different between families along an elevational gradient. And as I mentioned before, I'm using Symbiotic as my study system, where it has 6 different families: Heliconia, Symbiotic, Custaceae, Echinacea, Marantiaceae, and Mutaceae, that are present along this gradient. Third, if there is adaptation of heat tolerance for those species that have populations in more than 2 sites along the gradient. I worked in 5 life zones or sites along the La Selva Barber Transect in Costa Rica, which is the largest preserved elevation gradient in Central America. Here are the 5 life zones we worked on and the specific elevation we took samples from. I was able to determine the heat tolerance for Symbiotic by warming the leaves at 10 different temperatures and then measuring fluorescence. From this, we extracted T50, that is the temperature at which fluorescence decreases by 50%. For the results, I was able to measure 62 species that naturally occur along the 5 life zones of the gradient. The general pattern that I was expecting to see was that heat tolerance would match the maximum temperature that is present in each life zone of the gradient. This is in the lowlands, where the temperature is higher than the rest, to have the higher heat tolerance, and at the highest elevation over here, that has the coldest temperature, to have the lowest heat tolerance. Even though those are the expected results, the reality is very different. So we observe a totally different pattern, where heat tolerance increases with the increase in elevation. And the pattern is like this. Thermal safety margins is T50 minus the maximum air temperature. In the left paragraph, you can see that T50 data and the red dots are the maximum air temperature over here. From this, we get thermal safety margins in the right graph over here. When thermal safety margins values are close to 0, that means that they are in higher vulnerability to global warming. Here we can see that no matter the trend in heat tolerance, the lowland species over here presents the higher vulnerability to global warming than the other species along the gradient. This is because the heat tolerance of the lowland species are already very close to the temperature they experience during the day. The second question was if heat tolerance varies among families. This graph shows T50 in the y-axis and different families distributed by each life zone. Here you can see that the same pattern is repeated, where heat tolerance increases with increasing elevation by family. And the same pattern, of course. The third question is if there is adaptation of heat tolerance for those species that have populations in more than 2 sites. Here in the graph, you can see that heat tolerance in the y-axis and different life zones in the x-axis. The same pattern of increasing heat tolerance when increasing elevation is repeated as well. It's very interesting because I was thinking that the phylogenetic signal that has been tested before could explain the increasing pattern of heat tolerance on the gradient. But here, the population of species are changing as well, which could be driven by other physiological constraints, such as an increase in UV light, for example, or other stress types that change along the elevational gradient, and not the phylogenetic signal. Well, we can conclude that species are locally adapted to the life zones along the elevational gradient and the La Selva-Barba transect. That second, heat tolerance increases with an increase in elevation at the family level and at the population level. And finally, that tropical lowlands and gibberellid species are in higher vulnerability than any other life zone in the gradient. Finally, I want to acknowledge my funding sources, OTS, Heliconian Society International, the Explorers Club, and the Ecology and Evolutionary Biology Department at UConn, and to all the people involved in one way or another in this project. Thank you so much for hearing my talk.
Okay, well, thank you for joining in on this really wonderful session and for taking the time to view my talk. My name is Ben Taylor, and I'm an assistant professor in the Organismic and Evolutionary Biology Department at Harvard University. And today I want to talk a little bit about the interesting and often nuanced effects that nitrogen-fixing trees have on the process of tropical forest regeneration. So I want to start by noting just how incredibly important regenerating tropical forests are to the global carbon cycle. If we look at this really landmark study by Pan et al. In 2011, we can see these massive carbon sinks in regenerating secondary tropical forests. And we can see this across the world's tropical forests. So if we think about this at the global scale, in fact, we find that more than 40% of the entire world's forest carbon sink is accounted for by regenerating secondary forests in the tropics. So understanding what mitigates the pace of global climate change and what determines the sort of broader global carbon cycle it's really important to understand the processes that determine how quickly tropical forests regenerate from past human land use. Now, if we look at a regenerating secondary tropical forest, in fact, most often these forests are nitrogen limited because the quickly regenerating vegetative biomass requires a ton of nitrogen to photosynthesize and put on new carbon. Now, these forests can get nitrogen from a couple of different places. Often trees get nitrogen from the soil, but the preceding agricultural land use often depletes the soil nitrogen pool. So fortunately, there's another way that at least many members of these forests can get their nitrogen, and that's through a process called symbiotic nitrogen fixation. So some members of this community can fix dinitrogen gas into bioavailable forms that they then incorporate into their plant biomass. And as those leaves then eventually turn over and fall down to the forest floor, these nitrogen fixing trees serve as input valves of new nitrogen into the surrounding ecosystem. Now, this addition of new nitrogen into the surrounding ecosystem might suggest to us that these nitrogen fixing trees should facilitate the growth of these nitrogen limited trees right around them. And indeed, that is the most common intuition, and that has led to a very common practice in which people actively plant these nitrogen fixing trees in regenerating tropical forests to speed along the process of forest growth. But if we take a minute to think about this from a natural selection point of view, you might consider that it doesn't make a lot of sense for these nitrogen fixing trees to actively facilitate those neighboring plants such that those neighboring plants can grow bigger and shade out the nitrogen fixer itself, right? So another possible scenario is that this nitrogen fixer uses its fixed nitrogen to fuel rapid growth rates. It grows very tall itself and it shades out its neighboring plants. In that case, you might imagine these nitrogen fixers actually provide these really strong competitive effects and reduce the growth of neighboring trees right around them. So it's possible that nitrogen fixers could actually slow down the process of forest regeneration. And we really don't have good data out there to determine or to demonstrate what the net effect of nitrogen fixers is on tropical forest regeneration rates. So that's the question that we set out to answer. Do nitrogen fixers actually promote forest regeneration rates in the tropics? And this is a question that I've been working on with 3 collaborators, Doctors Robin Chasden, Duncan Mengi, and Bene Bachelot. And we set out to answer this question primarily at La Selva Biological Station, which is here in northeastern Costa Rica. La Selva is a great place to answer these questions because La Selva, if you look at this map over here, has a really patchwork landscape of forests of different ages, which are represented by different colors on this map. So what this allows us to do is set up a chronosequence of different plots of different forest ages that are still in close proximity to each other. And that's exactly what we've done with the Bosques project here. This is a set of chronosequence plots. Each plot is 1 hectare in size, and every single year, each plot is completely censused. All trees are tagged, identified, and measured, so we can calculate growth rates, and we can use the identification to determine which trees are nitrogen fixers and which trees are not. So we use these data to first ask this question at the plot scale. Do nitrogen fixers actually improve forest growth rates? Here on the x-axis, we've got the percent of the total basal area in a plot that's made up of nitrogen fixers. And on the y-axis, we've got the change in total basal area of the plot itself. So this is the growth rate of the plot. Now, if nitrogen fixers are good for forest growth rates, we would expect a positive relationship across this axis. When we actually looked at the data, though, we see the exact opposite result, that plots that have more nitrogen fixers in them tend to grow slower than plots that have fewer nitrogen fixers in them. Now, I'll say that it's possible that if nitrogen fixers themselves just happen to be slow growers, then we would see this pattern because more nitrogen fixers in a plot would just sort of bring down its average growth rates. So we then looked at just the growth rates of non-fixing plants or non-fixing trees in these plots. And that's what I'm showing you down here at the bottom. And in fact, we see the same pattern that plots with more nitrogen fixers in them, the non-fixers in those plots actually grow slower than in plots with fewer nitrogen fixers in them. Now, frankly, this was a pretty surprising pattern to us. So we wanted to really dig into that pattern more mechanistically at the individual scale to see how individual nitrogen fixers influence the growth rates of the trees around them. So to do this, we used something we call the neighborhood crowding index. And we looked at the amount of the neighborhood crowding index that comes from nitrogen fixers. So I'll explain this critical sort of x-axis here to you. So if we imagine a tree out there in the forest that has a set of neighboring plants that are crowding it, if those trees, neighboring trees, are non-fixers, then it's going to end up here on the left-hand side of this x-axis because 0% of its crowding is coming from nitrogen fixers. But if we imagine that same tree, it's got the same number of surrounding neighbors, those neighbors are the same size and the same proximity to it, but now all of those neighbors are nitrogen fixers, then it's going to end up over here on the right side of this axis. 100% of its crowding is going to come from nitrogen fixers. So all else being equal, if nitrogen fixers are good for their neighboring trees, then we would expect that the farther right you go on this axis, the higher the demographic process of that tree is, the higher growth rates of a tree are if more of its neighbors are nitrogen fixers. However, if nitrogen fixers are bad for their neighboring plants, we would expect negative relationships across this axis, as more nitrogen fixing neighbors means your growth rates, for example, are slower. Okay, so let's take a look at some of the data. Here we've got growth rates on the y-axis, and we've got this crowding from nitrogen fixers on the x-axis here. In fact, when we looked at all of the data in these Bosques plots, we see this strong negative effect of nitrogen fixers on the growth rates of individual neighboring plants. So for non-fixing plants out there, having all nitrogen fixers as your neighbors decreases your growth rates by about 40% relative to having all non-fixers as your neighbors. If you're a nitrogen fixer yourself, so you're represented by this red line, then having all nitrogen fixers as neighbors reduces your growth rates by about 60%. So these are actually really strong negative effects that nitrogen fixers are having on the growth rates of their neighboring plants. Now, we also see similar patterns for other demographic rates, recruitment and survival. We see either strongly negative or null effects of nitrogen fixers on their neighboring plants. In no cases did we ever find any strong positive or facilitative effects of nitrogen fixers on neighboring trees. Now, what's really fascinating or I think wonderful about this is that since this work came out in 2017, there have been several follow-up studies that have looked at similar effects of nitrogen fixers in other tropical regenerating forests. And we find a wide range of different results. So in Trinidad, we find positive effects of nitrogen fixers on forest growth rates. In Costa Rica, we find net negative effects of nitrogen fixers on forest growth rates. In several studies, we find effectively null effects of nitrogen fixers on growth rates. And then when you look at larger scales, either pan-tropical or across the entire coterminous United States, you see very context-dependent effects of N fixers on forest growth. So what this really suggests to me is that we need to take a much more nuanced approach to understanding how nitrogen fixers influence forest growth rates, especially in light of the fact that it is an extremely common practice to actively plant these nitrogen fixing trees in regenerating tropical forests to speed along that growth rates. These data suggest that's either not a good use of restoration funds or potentially even a counterproductive measure for sort of forest management in these regenerating forests. So with that, I just want to say thanks to all of my really wonderful collaborators, especially the Costa Rican team of census workers.
Thanks for our speakers and for the amazing presentations. And also we have a lot of great questions. We have very little time, so I'm gonna go straight to the questions and I'll go in order. Again, I apologize for any mispronounced name. From Flavia Sam to Scheherazade, very interesting work, nice approach. I was wondering how can community prevent hunting? Hola.
Thanks, Sophia, and thanks, Flavia, for your questions. I think for this particular village that we've been working on, so it used to be in the past, they allow hunters from the other areas to go to that village and hunt and take all the bats. But now, since we were in that village, and at first I stay on that islands with the local community and especially the fishermen groups to build trust and relationship and even engage them in the research itself. So I had the fishermen holding the bats for me, and they really get through the process with me together and understanding the bats and understanding what they actually feed, what kind of plants that they get a benefit from. And we've also worked with the community until the stage that the island where the bats are become the icon of the village. And it's some things that they've been really looking forward to have a vision together, to have these iconic islands as part of their village or community to work together for the ecotourism beside the conservation. So it's something that we're also working on. And yeah, I think that's the question, Sophia. That's the answer, Sophia.
Okay, awesome. Then you have some kudos also from Deep Chatterjee. His question is, what about the kind of awareness campaign you're carrying out? Are there any age group you are targeting, and what are the challenges you're facing while carrying out these campaigns?
Oh, okay. Yeah, thank you so much for that great questions. So most of the times that community-based conservation is not really deemed as has a strong scientific justifications, right? The scientific components. But given my background as the scientist itself, we design our awareness campaigns in certain way to target specific audience. So there are two main groups. First, the fishermen, and the seconds are actually the children's. So based on our assessment, the fishermen are the ones that actually at front, whether they want to carry the hunters to the bat islands to take the bats. And they're the ones that can decide, oh, yes, you can take or you cannot take the bats. So they were the first target group of our awareness campaign. We had a movie night watch, and we've not really in that formal awareness campaign, but more like informal discussions and ongoing conversations. And the second for children is mostly to also engage their parents, because once we have the children and hook in the conversations, the parents become more curious on what's going on around. And we've tested whether the children understand our awareness campaigns and so on. We developed from that durian study that published in Biotropica. We actually used the information to make the first that children book that we distribute in the village and the community. So that means a lot.
Awesome.
Thank you.
Thanks to you. Now from Brina Barlow to Georgia. The plants you studied are understory species. Do you think these shade-tolerant adapted species reacted differently due to their specific understory physiology?
Hi, Sophia. Thank you for the question.
Hola.
Well, actually, most of the species I'm working with are open area species. And something interesting that was happening was that we noticed that the species that were actually understory in the lowlands tend to live in like more open areas as you go up in the mountain. So that was very interesting. So that's why I thought probably it's UV light that it's having an effect on what we're seeing right now. But it's just like some things that we're like starting to think, nothing that we have data on right now.
Thanks, Georgia. Now for Bento Taylor from Antonio Crespo. Can nitrogen fixing trees be disregarded as facilitators of restoration?
Thanks. That's a great question, Antonio. I don't think so. I think that it's a context dependent effect is my best thought on it, and we still don't really understand exactly what the context is that makes them particularly strong facilitators or particularly strong inhibitors of forest growth. But I do think that it's at this point sort of a cautionary tale that it's not a blanket positive effect. I don't think it's either a blanket negative effect either, but sort of we need more information at this point.
Yeah, interesting. You can stay there for Benton too, from Shanpang. Fascinating insight into the role nitrogen fixers have on forest growth rates. Could there be some driver for the survival of nitrogen fixing trees that reduces the growth rates of trees in general? Perhaps nitrogen fixing trees have higher survival rates in poor soil conditions. Wasn't sure if this was accounted for, and I missed it.
That's a great question, and your intuition is correct in these plots. At least the nitrogen fixers that dominate in the plots that we work in do have particularly high survival rates, higher than the average non-fixers in these plots. So that does, in many ways, demographically drive their success. Now, that is inherently accounted for in the first figures that I showed at the plot scale, where there's total sort of plot scale change in basal area. That takes into account recruitment and survival or mortality. So the sort of lingering biomass in nitrogen fixers is with higher survival rates is accounted for in those. But I do think that, yes, survival, especially in in poor conditions, is potentially a strong reason why nitrogen fixers do really well in some places and might have particularly strong negative effects on their neighbors.
Okay, awesome. From Robin Chasden, do you think the positive or negative interactions, this is for Benton as well, between nitrogen fixing trees and neighbors is mediated by light availability and fourth stage of regeneration or restoration?
Yeah, that's a great question, Robin. We have obviously thought about this a lot together. Yes, in my mind, light is the big driver of this. That nitrogen, the ability of a plant to glean nitrogen in many ways allows it to fuel very rapid growth rates. And I think especially in these plots, those nitrogen fixers are particularly good at growing up relatively quickly and shading out their neighboring plants. So the inhibitive effects that we see of nitrogen fixers on neighbors, I think, are much more a story of other resources besides nitrogen. The fixers use nitrogen to get bigger, and then that depletes other resources like light availability for their neighboring plants, and that's why they have these strong negative competitive effects.
Awesome. Okay, I brought it the last one. Let's see. From Jessica Murray, Ben, have any other factors, soil, nitrogen content, sunlight, tree identity, etc., been observed to mediate these nuanced effects of nitrogen fixing trees on neighboring tree demographic rates in regenerative forests?
Yes, we have started to try to look at that, although the picture isn't completely clear or comprehensive yet. Apologies for moving outside of the tropics at this conference for a moment, but we've looked at a lot of these types of factors in the coterminous United States here with a different set of data, and we often see that that there's less competition when there's more nitrogen deposition, for example, at a site and that sort of thing, and when the nitrogen fixer and the non-fixer occupy more separate niches. So if the nitrogen fixer is, for example, a broadleaf species and the non-fixer is a needleleaf conifer. Which is maybe the nitrogen deposition story is maybe particularly surprising. It seems that the stronger, you might imagine that nitrogen fixers facilitate neighbors most in very nitrogen limited scenarios where the nitrogen that they fix can help their neighbors. But in fact, it seems like in nitrogen limited scenarios, the competition effect is particularly strong, particularly because nitrogen fixers themselves have unique access to this really important limiting resource. So that's what we have at the moment, but we're still looking into these concepts.
Okay, awesome. We have 3 minutes. Maybe we can answer the last one very shortly, which I now lost. Let's see. Here, okay. Is there, from AA, is there any research that looks at growth rate in regenerative forests as a function of herbivore density? My hypothesis is that mammalian herbivores might help add nitrogen to the system if they move from less depleted nitrogen areas of the forest. Fabio, from Fabio. That's for you, Ben.
I'm sorry, could you repeat it? I thought that one was for
of course.
For Jacob. I'm sorry. Where are we?
Let's see. Is there any research that looks at the growth rate in regenerative forests as a function of herbivore density? My hypothesis is that mammalian herbivores might help add nitrogen to the system if they move from less depleted nitrogen areas of the forest.
That's a good question. I do not know of it, if there is. So that's a fascinating question, and I agree with the intuition. I don't know of a data set that looks at that currently, but I certainly wouldn't count that as a comprehensive understanding of it.
Awesome. So there are many more questions, lots of them are really good ones. So we just want to remind you that the discussion can continue if you create a community or a meetup in Huova. And with these, we're gonna end the session. I remind you that there are lots of great Zoom backgrounds that you can add to your Twitter or Instagram handle, and you can download them at tropicalbiology.org. I'm gonna also copy in the chat the link where you can these. And then I want to thank everybody, all the speakers and all our attendees. Saxahavi, who has been in the back also, as is our Zoom tech. And all of this is very encouraging for lots of new scientists that are coming. So we hope to see you again, as I said, next year here. Thanks to everybody.
Thank you.
Thank you.